In Lecture 5, we are going to be looking at cellular respiration in a lot more details. We're going to look at how glucose is being broken down into carbon dioxide, and in the process, how you are going to be able to extract the energy to create ATP. Metabolism, you might remember from last lecture, refers to all the chemical process that takes place within a cell. Cellular respiration is probably more correctly defined as a metabolic pathway because it doesn't actually happen in a single step. We represent it in a single chemical equation, but it's actually broken down into many smaller steps, and each of these smaller steps, each of these chemical reactions are going to be catalyzed by a specific enzyme. So here it is again, the chemical equation for cellular respiration. We use glucose that we ingest from our food, and we react it with 6 molecules of oxygen, and oxygen would be something that we can obtain through breathing in the air, and in the process, after many steps, you will ultimately be generating 6 carbon dioxide molecule that you will be breathing out, as well as 6 water molecules that we will either keep in the body, or we can excrete it through urine output or sweat or other means. But the most important part is that this kind of reaction is a type of catabolic reaction, which means you're going to be able to generate energy by breaking down glucose, and we will try to capture all these energy that are being released to drive the production of ATP. So to recap, ATP stands for adenosine triphosphate, and here is a typical ATP molecule. You have your adenine, you have your ribose sugar, and 3-phosphate. So in this form, in the ATP form with 3-phosphate, your molecules is actually charged up. It contains the most amount of chemical energy, and that energy is going to be released when you hydrolyze the ATP into ADP and a phosphate group. That is, if you add water molecule to an ATP, you'll be able to break off the last phosphate, and that's going to release energy, the energy that you can now use to do mechanical work like moving a flagellum, transport work such as active transport, or chemical work such as catalyzing a chemical reaction. And here is just to show you the hydrolysis of ATP in another way. Right? We have the ATP here, and the bond that connects the last 2 phosphate is a high energy bond. So when you add water molecule to break that bond, tremendous amount of energy will be released, and again, you can use it to do various cellular work. And once used up, the ADP will need to be recharged in order for you to be able to use it again. So to charge this up, you will have to add energy to it, and that's what this lecture is about. We're going to talk about how breaking down glucose is going to be able to supply the energy necessary to recharge your ADP so that you can create ATP again. Okay, so this ATP and ADP thing, it's kind of like a rechargeable battery that is in the body all the time, and you know, every time you finish using the battery, you need to recharge it. Okay, and then you will drain it for using the energy, and then it will go back to empty again, and then you just keep on charging it, using it, charging it, using it. Okay, so that's called the ATP cycle. The cellular respiration reaction is actually classified as a redox reaction. Okay, so in a redox reaction, there are 2 reactions that are going on. There is the oxidation reaction, and there is a reduction reaction. So oxidation occurs when a reactant is like the ingredients, right? In a chemical reactions, they're shown on the left side of the equation. In an oxidation reaction, the reactant is going to be losing electron. Okay, so losing electron is going to be called oxidation. They are being oxidized. So you can remember Leo. Okay, loss of electron is oxidized, and on the other hand, if it's a reduction reaction, then the reactant is going to be gaining electron, and they are going to be reduced. Okay, so gain of electron is reduction, so that's GER here. So most people use the acronym, abbreviation rather, Leo and GER to remember which one is which. In the context of cellular respiration, we would have glucose being oxidized into carbon dioxide. What that means is glucose will be losing electrons. Okay, we will be stripping electrons away from glucose, and when you do that, the glucose will ultimately become carbon dioxide. And all those electrons that you strip from glucose will now be transferred onto oxygen. So oxygen will gain electron. It will be reduced into water. Okay, so because this reaction involves both reduction and oxidation at the same time, that's why it's called redox reaction. Okay, reduction and oxidation. Cellular respiration actually has 4 stages, and each stage is going to be broken down into many mini steps. Okay, so we have the first 3 stages shown here: glycolysis, pyruvate oxidation, citric acid cycle, also known as the Krebs cycle. So these 3 steps are going to be involving removing high energy electron from the glucose. Okay, so you would remove high energy electron from glucose, and eventually at the end of the 3rd step, at the end of the Krebs cycle, all the electrons from the glucose would have been removed, and then glucose would have been completely oxidized or broken down into carbon dioxide. So all these high energy electrons will be captured and be used in the last step. Okay, the electron transport chain, and in that last step, you will use these high energy electron to create ATP. So that's basically the premise of your of the cellular respiration and the 4 stages that are involved. Now, in order to move those high energy electrons that are released from the first 3 steps of cellular respiration, we need to have some kind of energy carrier. Okay, so these energy carriers are going to capture the high energy electrons released from glycolysis, Krebs cycle, and pyruvate oxidation, and we're going to transport them to the last step, to stage 4, electron transport chain, where you can harness the energy from these electrons. So there are 2 main types of energy carriers in the cell, in human cells. One of them is called NAD+, the other is called FAD. Okay, they are acronyms for something much longer. For the purpose of this course, we can just call them in their abbreviated form. Okay, so NAD+ and FAD will be capturing electrons from the first 3 steps, right? And then they are going to take these electrons to the electron transport chain, where you will be using them to produce ATP. So you can think of the NAD+ as a cap. Okay, when it's empty, then it's going to be called the NAD+. Okay, and the passengers are like the electrons. So the electrons will be coming out again from the first 3 steps of the cellular respiration. They will hop on to the taxi. They will combine with the NAD+, and once the electrons combine with the NAD+, we now call it NADH. Okay, so the NADH is the loaded form of the molecule, and here you can see that the NAD+ molecule actually combines with the electrons and one H+, producing the NADH. That's what the H is the H+, and it will create a free proton that's floating around. Okay, so this loaded taxi, this loaded molecule NADH will transport the electron to the destination, and the destination is going to be the electron transport chain. Once they get to the electron transport chain, the electron will hop off, and we will be able to use them to produce ATP. And more importantly, once the electrons got off, then the taxi, the NAD+, is free again to go back and pick up more passengers, right? The NAD+ can go back to the first 3 steps, get more electrons, and then it will shuttle them to the last step, the electron transport chain once again. So you can think of the NAD+ as well as the FAD as a mere a shuttle surface for the electrons, okay, so that they can go from the first 3 steps to the last step. Okay, so probably I should mention here, FAD is the other energy carrier. It actually carries less energy than NADH, and when they pick up the electron, this is going to get converted into FADH2. Okay, so this is the empty version, and this is the loaded version for FAD. So here is a roadmap for cellular respiration. We have glycolysis being the first step, and that happens in the cytoplasm of the cell. The product of the glycolysis is something called pyruvate that will then go into the matrix of the mitochondria, where it will undergo the second step, which is the pyruvate oxidation, as well as the third step, which is Krebs cycle. All this time, you will be releasing high energy electrons, and these electrons will be carried via the NADH. Okay, so the NADH and the FADH2 will take these electrons to electron transport chain, where you'll be producing ATP. Okay, you will also be producing ATP in glycolysis and Krebs cycle, but compared to the amount that you will generate in the electron transport chain, these are relatively small amount. They account for only about 10% of all the ATP production per glucose. Okay, so we're gonna look into each of these steps and see what exactly is going on, and what's being involved. So first, let's begin with glycolysis. The actual glycolysis is a 10-step process involving very complicated chemical names, and then there are going to be enzymes shown here in blue that help catalyze each of the reactions. And you can see you start with glucose, and the chemical structure is going to change along the way. So this is an introductory course, so we're not going to be going into all the glory details here in glycolysis. If you're interested, you can probably read that from a more advanced textbook, or do a Google search, but we're just going to stick with the basic of glycolysis. So glycolysis basically means the splitting of glucose, right? Glyco is the glucose and lysis is to break. So you are literally breaking the glucose into two. Right? So glucose is a 6-carbon molecule. When you split it into two smaller molecules called pyruvate, they will be 3 carbons each. Okay, so you're splitting a 6-carbon glucose into 2 3-carbons pyruvate. Sorry, the glucose is actually right here. That's just glycolysis. So you're splitting it into 2 3-carbon pyruvate. Like I said, there are 10 steps involved in glycolysis. Now, the first 5 steps are actually called the energy investing phase. All right, you might have heard of the phase where investors say it takes money to make money, right? So this is the same concept. It takes energy to make energy, right? You will have to first invest a little bit of energy in the first 5 steps in order to prep the glucose molecule for the reaction, and then you will be able to create more energy in the final 5 steps, which are called the energy yielding phase. Okay, so we have the investing phase for the first 5 steps, and once you've invested a little bit of energy, this is kind of like the payoff phase, where you will get those energy back and some more. Okay, so the energy that are produced in the last 5 steps will be in the form of high energy electrons, which will be captured by NAD+, the energy carrier, as well as some ADP. So once the NAD+ captures the high energy electron, they will become NADH. This is a typo here. I'm sorry. I'll change it in the PowerPoint that I post. It will be converted into NADH, which will shuttle the electron to the electron transport chain, and some of the energy will be used to add the phosphate group to ADP, therefore creating ATP. So here you can see we start with the 6-carbon glucose. You will first be investing 2 ATP. Okay, so this is the investment phase, the first 5 steps, and by investing 2 ATP, you energize the glucose molecule. You make it a little bit unstable, so it's more likely to react in the next 5 steps, and then the glucose will be split midway. Each of these molecules will undergo a series of chemical reactions, and along the way they will be releasing high energy electron, and you can see it's captured by the NAD+ here, as well as an NAD+ here, and then later on it will be releasing even more energy, and that energy is used to add a phosphate group to the ADP, creating ATP. So you can see in this diagram, we invested 2 ATP at the beginning. Okay, so minus 2 ATP at the beginning, and at the end we actually get plus 4 ATP. Okay, so by investing 2 ATP, you end up having 4 ATP. So the net ATP production is going to be 2. Okay, 4 minus 2, right? Okay, so finally you will get your 2 pyruvate. Sometimes they call it pyruvic acid. It's the same thing. Okay, pyruvate is just that when you remove a hydrogen from pyruvic acid, then it becomes pyruvate. It doesn't really matter for our purpose. What's important is you have to know that the pyruvate is a 3-carbon molecule that you get when you split glucose into 2. So these ATPs here, right? How do you actually produce those ATPs? Well, there is something called substrate-level phosphorylation. Okay, phosphorylation. To phosphorylate something is to add a phosphate group to that molecule. Okay, we can actually add phosphate groups to many things. You can add it to a protein, you can add it to an organic molecule, and in the context of what we're talking about, we are going to be adding it to the ADP, so that you will get ATP. Okay, so in substrate level phosphorylation, you're going to need 3 things. Right? You're going to need an enzyme to catalyze the reaction. You are going to need to have a fuel molecule. Okay, a fuel molecule is a molecule that will be donating a phosphate group. So over here we have 3 carbon molecules with 2 phosphate group. Okay, and the third thing that you need will be ADP. So in the reaction, the enzyme will take the phosphate group from the donor molecule, from the fuel molecule, and transfer it onto ADP. And the result of that is you will have ATP, right? And the molecule will now have 1 less phosphate. Okay, so let me just go back here. You can see that there is a 3-carbon molecule with 1 phosphate, and then in a separate reaction, this molecule gets phosphorylated, meaning you're going to be adding a P to it. That's why it ends up having 2 phosphate. And then what happens is in substrate level phosphorylation, this molecule will be donating its phosphate group to 2 separate ADP, therefore generating 2 ATP. Okay, and so these 4 ATP in glycolysis, they are all produced by this method called substrate level phosphorylation. As mentioned earlier, substrate level phosphorylation is not the main method of creating ATP. It only accounts for roughly 10% of all the ATP that you are going to be produced. So here is a summary of what happened in glycolysis. First, it takes place in the cytoplasm of the cell, and the glucose will be broken down into 2 pyruvate molecules. In the process, you will be releasing energy that will be creating 2 net ATP. That's after subtracting the initial 2 ATP that you've invested, and the high energy electrons released along the way will be captured by NAD+, generating 2 NADH. Okay, so we will take a look at the fate of these NADH later on when we get closer to the to step 4. But now let's take a look at what will happen to pyruvate. Okay, the fate of pyruvate will actually be dependent on the type of cells that we are talking about. It's going to be different for a human cell. It's going to be different for, say, a yeast cell. Okay, so different organisms will have different pathways for pyruvate, and it will also depend on the environmental condition. Even within the human cells, whether you have sufficient oxygen or not, that would cause pyruvate to go in 2 separate paths. So with sufficient oxygen, the pyruvate will now undergo aerobic respiration, and it will enter the mitochondria to do phase 2, stage 2. If there is not enough oxygen, such as during strenuous exercise, then the pyruvate will undergo anaerobic respiration instead. Right? So aerobic means with oxygen, anaerobic means without oxygen. Okay, and so situation will cause the pyruvate to undergo fermentation, which we will look at the end of the lecture. But now we're going to focus on aerobic respiration. Okay, we'll look at what happens to pyruvate when there are sufficient supply of oxygen. So stage 2 after glycolysis is going to be something called pyruvate oxidation. And oxidation, as we mentioned earlier, means a loss of electron, right? So you are going to take more electrons away from the pyruvate. So there are 3 separate steps that are involved in the pyruvate oxidation. First, we will remove a carbon dioxide from pyruvate. Okay, so remember, pyruvate is something that you'll get when you split the glucose in half. So this is a 3-carbon molecule. And when you remove a carbon dioxide from it, it will now become a 2-carbon molecule called acetic acid. Okay, so pyruvate becomes acetic acid after you remove a carbon dioxide. Then you are going to oxidize the acetic acid. Okay, and you're going to remove electrons from it, and those electrons that you remove, as always, are going to be high energy electron, and we want to capture them. Okay, we want to take them to the electron transport chain, and in order to do that, we need the energy carrier NAD+. So the empty taxi comes, picks up the passenger, and now this is going to go to the electron transport chain, or the ETC. Yeah? And because we have been stripping electrons from these molecules, so the energy level might be a little bit lower now, and the molecules are kind of stable, and it might not want to react further. So to make sure that the molecule will continue to react, we're going to give it a little bit of a jolt. Okay, we're going to give it a bit of a boost of energy, and to do that, we will combine it with an energetic molecule called coenzyme A. Okay, so coenzyme A will bind to acetic acid, and it will create a high energy unstable bond in the molecule, so that the molecule is going to be primed for the next stage of cellular respiration. Okay, so after the coenzyme A binds to the acetic acid, you will generate this molecule called acetyl-CoA. Okay, so acetyl-CoA has been energized by the coenzyme A. It's now ready to react a little bit more. It's important to note that from glycolysis, you actually get 2 pyruvate. So everything that I just described here actually occurred twice. So you'll be getting 2 carbon dioxides per glucose. You'll be getting 2 NADH per glucose, and you'll be getting 2 acetyl-CoA per glucose. Okay, so this is 2 pyruvate acid or pyruvic acid or pyruvate coming in, and you'll be generating 2 of each of these things. So here's a summary of the 3 steps that are going to be involved, right? Removing the carbon dioxide, that is called decarboxylation, and then you have high energy electron release and captured by NAD+, and finally we are adding the coenzyme A to the pyruvate so that you are going to be creating the acetyl-CoA. Here is a summary of what happens in pyruvate oxidation. The pyruvate that was created from the glycolysis will enter the matrix of the mitochondria, and in the matrix, that's where the pyruvate oxidation will occur. And just in case you forgot where the matrix is, right? So this is the mitochondria, and then there is the inner membrane of the mitochondria, and the fluid inside there. That's where the matrix is. Okay, so the matrix is where the second step will occur. In this step, pyruvate will be converted into acetyl-CoA, and for every glucose you're going to get 2 acetyl-CoA, because 2 pyruvate will be going into this step, and you will be generating 2 carbon dioxide as well as 2 NADH. So that's it for this video. In the next video, we will look at what happens in a Krebs cycle as well as the electron transport chain.